All right, let's dive right in. Today we're talking about something that has totally transformed biology, next-generation sequencing. This is the story of how we went from, slowly trying to decipher the code of life one letter at a time to reading entire genetic encyclopedias in just hours. And trust me, this isn't just a small step forward, it was a full-blown revolution. So I want to start with a number, 13 years. Just think about that for a second. What kind of massive, global, scientific project would take more than a decade of constant work from thousands of brilliant people to get done? And what if I told you that on top of those 13 years, it also came with this incredible price tag, $2.7 billion. We're talking about an absolutely monumental effort. So what on earth was it? Well, that was the time and money it took to create the very first complete map of the human genome. It was, without a doubt, a historic achievement. But its insane scale made one thing crystal clear. We desperately needed a better, faster, and way, way cheaper way to read DNA. The biggest problem was the method they were using. I mean, the early way of sequencing was completely linear. Imagine trying to read a book with 3 billion letters, but you could only identify one single letter, then the next, then the one after that. It worked, sure, but it was just painfully slow and ridiculously expensive. So to really unlock all the secrets hidden in our DNA, not just for one person, but for everyone, we couldn't be spending billions of dollars and waiting over a decade every single time. Science needed a massive breakthrough. And thankfully, it got one. A new technology that just changed the entire game. The original method was called Sanger sequencing, named after the guy who pioneered it, Frederick Sanger. It was brilliant for its time, no doubt, but like we said, its biggest limitation was that it could only read one piece of DNA at a time. That one-by-one approach was the ultimate bottleneck. And then came next-generation sequencing, or NGS. The absolute genius of NGS is this idea called massive parallelization. Instead of reading one DNA fragment, the question became, why not read millions or even billions of them all at the exact same time? It's like going from one person reading a book to an entire stadium of people all reading different pages simultaneously. Okay, this slide really puts it all into perspective, doesn't it? The old way, Sanger sequencing, gave you really low throughput. It was slow, and that meant the cost was just astronomical. NGS completely flips that on its head. The throughput is huge, the speed is mind-bogglingly fast, and because of that, the cost per genome just plummets. It's a totally different way of thinking, but the real game changer here is that scale. Doing this for millions of strands at once was something that was pure science fiction just a few years before. So how in the world does this massive parallelization thing actually work? Well, the best way to picture it is to think of a super high-tech factory, one that's designed to process billions of tiny little DNA blueprints all at the same time. It really boils down to this 4-step process. First, you take the long DNA strands and just shred them into millions of short, easy-to-read pieces. Simple enough. Second, you attach these special little chemical tags called adapters. Think of them like barcodes that also help anchor the DNA fragments down. Third, the machine does its magic and reads all of these millions of fragments at the same time. And finally, you have this incredibly powerful software that acts like a giant puzzle solver, taking all those short reads and piecing them back together to recreate the original genetic sequence. And that brings up a really key point. This wasn't just some biology innovation. No way. This was a triumph of teamwork, of combining biology with super high-performance computing, advanced optics, and even microfabrication. It really took experts from a whole bunch of different fields to build this DNA factory. Okay, so you've done it. You've sequenced millions of DNA fragments all at once. That's great. You've solved one massive problem, but you've just created a brand new one, a literal tsunami of data. What do you do with all of that information? Well, the answer comes in a standard file format called FASTQ. It looks a little cryptic, but it's pretty straightforward. For every single little piece of DNA that gets read, the machine spits out 4 lines of text. You get a unique ID, the actual DNA sequence itself, the A's, T's, C's, and G's, a little placeholder line, and then, most importantly, this 4th line of jumbled characters, which is the quality score. And that quality score, or Q-score, is absolutely crucial. It's basically a confidence rating. For every single letter the machine reads, it assigns a score that tells the scientist, hey, how certain am I that this is the right letter? It's the machine's way of saying, yeah,
I'm 99.9% sure this is a G.
This chart gives you a sense of what those scores actually mean. Now, a Q score of 20 is already 99% accurate, which sounds pretty great, right? That's about a 1 in 100 chance of an error. But in genetics, that's not quite good enough. The real gold standard for high-quality research is a Q score of 30. That gets you to 99.9% accuracy, only a 1 in 1,000 chance of a mistake. That's the level of precision you need to really trust the results.
All right, let's get to the payoff. We have this unbelievable technology that's fast, it's cheap, it's super reliable. So what? What can we actually do with it? How is this changing our world right now?
One of the biggest areas is personalized medicine. We can now affordably read a person's entire genome, which unlocks all these insights that used to be hidden inside that billion-dollar map. Doctors can spot tiny genetic variations, they're called SNPs, that are linked to diseases like cancer. They can predict if a certain drug will work for you or help diagnose a rare genetic disorder that's been a mystery for years. It's the beginning of medicine that is truly tailored to you as an individual.
But it goes so far beyond just human medicine. This revolution lets scientists map the genetic code of, well, our entire planet. They're using NGS to piece together the genomes of newly discovered species, to understand a whole ecosystem just by sequencing DNA from a scoop of soil or water. They can even see which genes are being turned on and off in our cells. It's become an essential tool for almost every part of modern biology. So let's just step back and look at this timeline. It's wild. From the invention of sequencing in 1977, it took all the way until 2003 to get that first multi-billion-dollar human genome. Just a few years later, NGS machines hit the market. And now, today, we can do that exact same job in about a day, for less than $1,000. The speed of progress is just absolutely breathtaking. So here we are. We can now read the book of life with a speed and a clarity that was unimaginable just a short time ago. This power opens up an entire universe of possibilities. And it leaves us with one heck of a question. What story are we going to choose to write next?